Okay. Hi everyone. A very good morning to Doctor and my fellow friends. And we are from Group 6. The topic for today is Dermatophytosis, Microsporum Canis. My group consists of me, Sri Livashni, and another one is my friend Mindy. So, introduction. Microsporum canis is a zoophilic dermatase fungus impacting the characterized tissues, example like skin, hair, nails in animals and humans. It is a major cause of dermatosis, ringworm in a veterinary medicine. It always cats are the main disorder, affecting both dogs and also cats with sympathetic and asymptomatic carriers. Transmission occurs through direct contact with infected animal or contaminated items, such as bedding, furniture, grooming tools. Zoonotic nature poses public health risks in environment with close human-animal interaction, example, veterinary clinics, shelters, and also the households. The clinical signs that include multifungal alopecia and scaly circular lesions resembling other dermatophyte infection. Last not least, the pathogens involve secretion of keratinolytic enzymes, example like keratinase, and activation of genes related to keratin degradation. Type of sample, they do have three samples, which has sampled from the active age of the lesion because it's the highest fungal activity. And secondly, skin scraping from lesion, margin, or cross material for the chronic lesions. Third, nail clipping from the affected nails. Next. Sampling techniques for the first, it uses Mackenzie toothbrush technique. It's a vigorous brushing over lashes for 5 to 7 minutes using a sterile toothbrushes. Carpet method. Pressing sterile polyester carpet square onto the affected skin to collect the hair and scales. Skin scraping collected using a sterile scalpel blades from the lesion at the edges of the lashes. Hair plucks using a sterile forceps to collect the broken or easily removable hairs. And lastly, nail samples. It is taken from the nail plate, nail bed, or sublingual region using a nail clippers. In how do they transport and storage the sample in the laboratory? They will store the sample in sterile, airtight container, example in petri dishes or dermatophyte kits. Transport the sample to the lab within 12 to 24 hours at the room temperature to avoid refrigeration or freezing. The lab sample should be written there, the location, the date, and then animal ID. If immediate processing is not possible, store the sample dry at the room temperature away from the moist and sunlight area. Next. Direct microscopic examination, the procedure, add a drop of 10 to 20% of potassium hydro to dissolve the keratin and clear the sample at the background. And dimethyl sulfite can be added to speed up the clearing, and heat the slide gently for 5 to 10 minutes to allow the cooling before placing over the cover slide. And then microscope observation, use a magnification of at least 10 to 40 times and look the fungal animal like elements like arthrospore, branching septa, hypa, and also spherical spores around the hair scarves. And optional stain, it can be used lactopanel cotton blue for the enhanced visibility. Fungal culture, the media, it can be used Saporal dextrose agar with Clomba panicle and cyclohamidae to inhibit the bacterial growth and other fungi. BBL mycocell agar with a panel red as a color indicator. Incubation condition, the temperature should be 25 to 30 cells in the warm and moist environment. The duration can be typically incubated for 14 to 21 days. Observation and begin after the 7 days. So if you could see, they do have pictures of the media and how do the colony can be appear. And the colony categorized can be spindle-shaped micronidia with a thick, rough walls and multiple septa observed through microscopically using a leptonok cotton blue stain. And microconidia a few and appear along the hyphae. So if you can see at the another picture, it can in the leptophenol cotton blue stain that you can observe the microscopically the micronidia with those few appear along the hyphae. Next.
Okay, so next is the macroscopic examination of native cultures. For the slide preparation, the sample placed on glass slide using stain of 10% KOH, potassium hydroxide, and it's helped dissolve keratin and reveals fungal element. And the second stain is lactophenol cotton blue (LPCB). It helps stains fungi color of blue. And then the microscopy is examined under 10 times and 40 times. And key features observed is septate hyphae and macroconidia, as the picture shown, and spores. And this purpose of microscopic examination is to aid in identifying dermatophytes and diagnosing fungal infections. And this picture shown is the club-shaped macroconidia. Next is the making slide cultures according to riddles. So the slide cultures with riddle methods is allowing fungi to grow directly on a slide for easier observation of natural structure. And the sample is placed on agar with a sterile slide, incubated at temperature range of 25 until 30 degree, and it's the ideal condition for the growth of most dermatophytes. Um, the stain using is lactophenol cotton blue, and this stain will helps highlight the hyphae and conidia for accurate fungal identification under microscopes. Next is the identification results. Microsporum canis was identified as the causative agents of dermatophytes through direct microscopic examination and slide culture. The characteristic spindle-shaped microconidia with thick row wall were observed. This dermatophytes commonly affect skin and hair, and it's typically transmitted from infected animals, especially cat and dogs, to human. As the picture shown, Microsporum canis dermatophytes. The picture A is the macroscopic identification of Microsporum canis dermatophytes, grown at 23 degree on SDA with a filamentous form. It raised center and flat periphery, has white to cream color. And the B1 is the macroscopic identification of Microsporum canis dermatophytes. It's numerous spindle-shaped macroconidia with 5 until 10 cells. That's all from us. Thank you for your
Thank you. Thank you, guys.